Good morning to one and all. Welcome to SR PHARMA GYAN. Today in our second lecture of glucagon and insulin, we are going to discuss about the insulin. Isolate of Langerhans of pancreas consist of the different cell like alpha cell, beta cell, gamma cell, delta cell, epsilon cell. Among them, almost 60 to 75% of cells are beta cell, which is responsible for the secretion of insulin. So insulin is secreting from the pancreatic beta cell. Now if we are talking about the synthesis of this insulin, what we can find that insulin is secreted from the pre-proinsulin to proinsulin, and then it will convert into the insulin. So first we are going to discuss about the synthesis of the insulin. In rough endoplasmic reticulum, majorly pre-proinsulin synthesis takes place. Now if we draw the diagrammatic structure of the pre-proinsulin, what we can find that here pre-proinsulin, it is consist of mainly four parts. So what are the mainly four parts? First one is the N-terminal A peptide, clear, a single peptide, clear, N-terminal peptide, which is composed of 24 amino acids. So this is the N-terminal peptide which is composed of 24 amino acid. Then this is the 30 amino acid one chain, which is the actually A chain of insulin. So A chain of the insulin is the second part, which is composed of the 30 amino acid. Next part, that is the 35 amino acid, that is nothing but the C peptide. So this part, it is the C peptide. And last part, that is the carboxylic acid, that particular N, it is the B chain of the insulin, which is composed of the 21 amino acid, clear, 21 amino acid. So pre-proinsulin, it is composed of the 110 amino acid molecule, clear, pre-proinsulin, it is composed of the 110 amino acid molecule. Now this pre-proinsulin, it will be transported from endoplasmic reticulum to Golgi apparatus, and in that process it will undergo the proteolytic breakdown. Clear, it will undergo the proteolytic breakdown, and as a result of the proteolytic breakdown, a single sequence peptide, clear, a single sequence peptide, that means N-terminal peptide, this one, N-terminal peptide, it will come out. There will be the cleavage, breakdown, clear, breakdown of the N-terminal peptide, and as a result there will be production or the synthesis of the proinsulin, clear, there will be the synthesis of the proinsulin. Now if you see about the proinsulin, that is B amino acid, which is consist of the 30 amino acid, then C peptide, which is composed of the 35 amino acid, and the next is the B chain of the insulin, which is composed of the 21 amino acid. So in together there is a 86 amino acid molecule, clear, an amino acid molecule it is there in the proinsulin. Clear, in the proinsulin there is a presence of the 86 amino acid molecule. And in this here we can also find that presence of the disulfide bond. Now in the disulfide bond, if we are seeing the disulfide bond in the A chain, if we are considering this is the A chain, clear, in the A chain, then here A6 and A11, in between the A6 and 11, this is the amino acid molecule, clear, molecule A6 and the A11 number, there is a presence of the disulfide bond, that is inside the chain A only, and another 2 disulfide bond it is present which is attaching the A chain and B chain of the insulin. Clear? A chain and B chain of the insulin, and here we can find that here the A7. Clear? A7, that means in the A chain 7th position amino acid and B chain 7th position amino acid. So in between the 7th position amino acid of the A chain and 7th position amino acid of the B chain, there is a disulfide bond. Another disulfide bond we can find in between the 20th position of the A chain and 19th position of the B chain. That means 20th position that amino acid and 19th position that amino acid of the B chain, there is a disulfide bond. So this is the pro-insulin. Clear? This is the pro-insulin. Now further, pro-insulin undergo proteolytic breakdown. This pro-insulin further it will undergo the proteolytic breakdown, and this proteolytic breakdown it will be occur in presence of pro-hormone convertase enzyme, that is the PC1 and PC2, pro-hormone convertase enzyme, and carboxypeptidase E, and in presence of carboxypeptidase E. So in presence of that there will be the proteolytic breakdown, and whenever there is a proteolytic breakdown, this C peptide, clear, this C peptide it will be get separated. Clear? This C peptide it will be get separated, and insulin it will be get separated. Clear? Insulin it will be get separated. So here you can find that C peptide which is composed of the 35 amino acid, and insulin it is composed of the 51 amino acid. Clear? 51 amino acid. Among the 51 amino acid, A chain composed of the 30 amino acid and B chain composed of the 21 amino acid, and this will be attached, A chain and B chain it will be attached with the 2 disulfide bond. Clear? 2 disulfide bond. So that is the synthesis of insulin. Clear? Hope this is clear to you. Next we are going to discuss about the release of insulin. Clear? Release of insulin. Now in case of the release of insulin, if we plot a chart, insulin release mg per ml versus time, what we can find that here you can find that this one, this is the normal condition insulin secretion. So this is the normal condition. In the normal condition insulin secretion. So in the normal condition we can see that insulin it is released as a biphasic release. There is a biphasic release. Now in the biphasic release, in the first phase the peak, clear, we can see that peak into the biphasic response, but it is the short life. Clear? It is the short life. In the second phase it was delayed but long life. Clear? It is delayed, long life means long duration of action it will be there. So it is the first phase and followed by the second phase. In case of the diabetes mellitus, clear, in case of the diabetes mellitus, here you can find this, see, type 1 diabetes mellitus means absolute deficiency of the insulin due to the destruction of the beta cell, absolute deficiency of the insulin. So insulin level you can find that there is a no peak, insulin level is very less. So that is the type 1 diabetes mellitus. In case of the type 1 diabetes mellitus, insulin level it is like that. In case of the type 2 diabetes mellitus, generally type 2 diabetes mellitus can be occur due to the insulin resistance or reduced insulin secretion. So in general, in case of the type 2 diabetes mellitus, we can see this type of the graph. Clear? This type of the graph where there is a diminished first phase. Clear? There is a diminished first phase but early second phase we can observe. So this is the release of the insulin or biphasic release of the insulin. Next we are going to discuss about the release of insulin. Now whenever we are taking the food, from the digestive system, intestine, food, there will be the digestion of the food and then it will enter into the circulation. Similarly, from the liver, glucogenolysis or the gluconeogenesis, through this process also in the blood there will be the glucose, amino acid or the fatty acid, it will be present into the blood. Now glucose, it will enter into the pancreatic beta cell. Clear? It will enter into the pancreatic beta cell through a transporter which is known as a GLUT. Now this glucose, this is the pancreatic beta cell. Clear? This is the beta cell of pancreas, so it will enter into the beta cell through GLUT. Now GLUT, there is a different type of the GLUT is there. Majorly GLUT and HGLT, these are the two major glucose transporters is present in our body. Now in the GLUT, in the pancreatic beta cell, that is the GLUT2. Clear? GLUT2, there is a different types of the GLUTs are there. This is the transporter. So glucose, it will enter into the pancreatic beta cell through GLUT2. Now after entering into the beta cell, it will undergo different you can say that conversion, glucose. Now through the different glucokinase, it will convert into the glucose 6-phosphate. So glucose conversion from the glucose to glucose 6-phosphate. Now this glucose phosphate, through the different steps, clear? Through the different steps, like it may be the glycolysis, citric acid cycle, oxidative phosphorylation, it will generate the ATP. Clear? It will going to generate the ATP. So this is you can say that that's why we are telling the ATP is a energy and it is converting from the glucose. Now the pancreatic beta cell, they consist of the two ion channel, majorly two ion channels. One is the ATP sensitive potassium channel, ATP sensitive potassium channel, another is the voltage gated calcium channel. So whenever glucose is entering into the pancreatic beta cell, so production of ATP it will be rises. So production of ATP it will be rises. So ATP production is increases. And this channel is ATP sensitive potassium channel. So whenever there is a increase into the you can say that ATP, then what will happen? Potassium channel it will be get closed. Clear? It will be get closed. Potassium channel it will be get closed. Whenever this channel it will be get closed, what will happen? The potassium cannot go outside. Clear? Potassium cannot go outside. So potassium is a positively charged ion. So intracellular potassium level it will be rises. Clear? There will be the rise into the intracellular potassium level. Whenever there is a rises into the intracellular potassium level, that is nothing but the depolarization. Now due to this depolarization, this voltage gated calcium channel it will be open. Voltage gated calcium channel it will be open, and through this voltage gated calcium channel, calcium it will be entered. Clear? Calcium it will be enter inside the pancreatic beta cell. So inside the beta cell, calcium level is rises, level of calcium is increases. So this calcium it will act on the granules which consist or which is containing the insulin, and through the exocytosis, clear? Through the exocytosis, insulin it will be released. Clear? Insulin it will be released. So by that process, glucose is responsible for the release of insulin from pancreatic beta cell. Now we are going to discuss about the various effects of the insulin. Now if you see about the various effects of the insulin in liver, if you are coming from the liver, in liver it is responsible for the decrease of the glycogenolysis. Clear? It is responsible for the decrease of the glycogenolysis. Similarly, in liver it is, of course you can say that it will reduce the conversion of amino acid to glucose. So it is responsible for the decrease of amino acid to glucose, that conversion. In liver, insulin is responsible for the decrease in the conversion of fatty acid and amino acid to keto acid. So that means fatty acid and amino acid to keto acid, that conversion also it will be inhibited. But it enhances hepatic glucose uptake, that means from blood to hepatic cell, hepatocyte, the glucose uptake it will be increases, so that blood glucose level it will be reduces. Then it will also promote the glycolysis and glycogenesis. Now in the glycolysis, it is nothing but a metabolic pathway which converts glucose to pyruvate and hydrogen ion, where in case of the glycogenesis, it is nothing but the synthesis of the glycogen, clear, glycogen. So it is promoted in liver. Then triglyceride and VLDL synthesis, clear, triglyceride and VLDL synthesis also it is enhanced in the liver. So these are the effect of the major effect of the insulin in liver. Coming to the muscle, so again muscle also, in the muscle also glucose uptake, blood to muscle glucose uptake, it will be promoted by the insulin, it is promoted by the insulin. Glycolysis and glycogenesis also promoted in the muscle by insulin. Glycogenolysis and glycogenesis. Increase protein synthesis and amino acid uptake. So in muscle it also promotes the amino acid uptake and synthesis of the protein. It restrain flow of alanine, lactate, pyruvate to liver, clear, to pyruvate to liver. Next is in adipose tissue. If you are talking about the adipose tissue, glucose uptake, it enhances the glucose uptake, clear, it is enhances the glucose uptake. It is enhances in the adipose tissue, it is enhances the glycerol synthesis, clear, glycerol synthesis, but flow of glycogenic precursor it will be reduced, clear, flow of glycogenic precursor it will be reduced. Now here muscle also we have told about the alanine, lactate, pyruvate. So this is nothing but the glycogenic precursor. These are the glycogenic precursor. Next is in adipose tissue, decrease into the lipolysis, breakdown of the lipid also inhibited or reduced in the adipose tissue. Next is in the triglyceride and fatty acid synthesis is promoted in the adipose tissue. So triglyceride and fatty acid synthesis it is increased into the adipose tissue. In brain it is reduce the appetite and increases the energy expenditure. So it is reduce the appetite and increases the energy expenditure. So this is all about the different functions of the insulin, through which it is maintaining the blood glucose level, that means glucose homeostasis, or we can say that it is reduce the blood sugar level and maintaining the glucose homeostasis. Now we are going to discuss about the insulin receptor. Now if we look into the insulin receptor, we can find that there is a two subunit of the insulin receptor it is present. One is the alpha subunit, which also called as a ligand binding site, that is present outside the membrane, clear, outside the membrane. So this is the alpha subunit, which is the ligand binding site, and molecular weight is 1,30,000. This is the cell membrane, and in the cell membrane you can say that intercellular beta subunit is present. Here it is the beta subunit. Now beta subunit is the intracellular signaling subunit. We can call it as a intracellular signaling subunit. Why? Because there is a presence of the tyrosine kinase domain. There is a presence of the tyrosine kinase domain. Now here if we are talking about that, molecular weight of the beta subunit is 90,000. Now here we can find that alpha subunit and beta subunit. In the beta subunit, that is the two insulin binding site is there, two insulin binding sites are there, and here we can find that tyrosine kinase domain. Now this is the tyrosine kinase domain, and it is the family of tyrosine kinase. It Clear? It is the family of tyrosine kinase. So insulin, it will bind to this insulin receptor. Clear? It will bind to the insulin receptor and produce different effect whichever we have discussed. Now insulin, whenever it will bind to this insulin receptor, it will follow majorly two pathways. So it will produce its activity via two pathways. So what are the two pathways? One is the PI3K ATK pathway, PI3K phosphoinositide 3 kinase and AKT pathway. This is the one of the pathway. Another is the RAS and MAPK pathway. Clear? RAS and MAPK pathway. So renin angiotensin system and mitogen activated protein kinase, mitogen activated protein kinase. So they will act by these two different mechanisms. Now we are going to discuss about these two pathway, how the insulin produce its effect. When the insulin bind to the insulin receptor, this tyrosine kinase domain, it will be undergo the phosphorylation. Clear? It will undergo the phosphorylation, and whenever there will be the phosphorylation, it will takes place, insulin receptor substrate or IRS, it will undergo phosphorylation and thereby the IRS1. This IRS1, then it will convert into the PI3K. It will activating the PI3K, and followed by there will be the activation of PIP2, phosphoinositol biphosphate and phosphoinositol transphosphate, PIP2 and PIP3. Then there will be the activation of AKT. AKT is nothing but it is the protein kinase B, AKT here. Now this AKT, again from the that is the eNOS and the NO synthesis, it will be takes place, but this AKT, it is mainly responsible for the translocation. Clear? Translocation. There is a different translocation proteins are there, like ASC160 is there. So they will be promoting the AKT is responsible for the promoting the translocation. Translocation of what? Here there is a GLUT4, it is present. Clear? Here it is present the GLUT4. In pancreatic beta cell we have discussed about the GLUT2, but here it is the GLUT4. So through the GLUT4, it will, you can say that, cause the uptake. Clear? It will cause the uptake of glucose. Clear? Uptake of glucose. So this process, there will be the uptake of the glucose or the gluconeogenesis, the muscle and fats, it will be, you can say that uptake of the glucose, it will be all those things, it will be processes. So this is about the PI3K AKT pathway. Another pathway, RAS and MAPK pathway. So in that case, whenever the insulin, it will bind to the insulin receptor. Clear? When the insulin, it will bind to the receptor, there is a different proteins it is there, like SHC, GRB2, SOS, this will be going to activate sequentially, and then there is a activation of the RAS. Clear? RAS. Then RAS2, activation of the RAF, then MAC, and MAC2, again you can say that MAPK, all those activation, it will be there, and then ERK activation, all those things, it will be happening. Now through this process, and then you can say that, of course you can say that if this activation is there, next is transcription, genetic transcription, and protein synthesis, it will take place. So this process, it will be important for the cell growth, gene expression, and protein synthesis. So this RAS and MAPK pathway, it is mainly responsible for the cell growth, gene expression, and protein synthesis. So this is the mechanism how the insulin produce its effect. So this is all about the insulin. Hope you have understood it. If you have any doubt, you can put your comment in the comment box. Thank you.